Hello students, today's topic is molecular aspect of speciation and speciation genes. Species are defined as a population of organisms capable of interbreeding among themselves, thereby sharing their gene pool through mating and genetic recombination. The genes are shared among the species. Individuals of a species are unable to mate with others of a different group because of gene barrier, as discussed earlier. The discovery of DNA as genetic material made it possible to compare the genome of distantly related species. The comparative studies enabled to give information about DNA and protein variations and the extent of variations, providing an evolutionary relationship among species. The study of how and why their sequences change through time constitute molecular evolution. New genes evolve through duplication and divergence. One of the best studied example is of the hemoglobin multigene family. The hemoglobin family probably descended from an ancestral globulin gene, myoglobulin. Occurrence of duplication followed by mutation and transposition lead to formation of alpha globulin family and beta globulin family. Changes and mutation occurred with time in the amino acid sequence of these hemoglobin polypeptide chains and led to evolution. The certain terms that are associated with this are homologous sequence. These are sequences that have common ancestor. Example, the hemoglobin alpha and beta gene have a common ancestry, the myoglobin gene. Orthologous gene. These are homologous genes that are from different genome. They retain the same function. Example, the alpha hemoglobin gene in frog, human and mouse. Paralogous genes. These are duplicated in the genome of the same species and acquire new or specialized function. Example, alpha globulin and beta globulin genes in mouse. Paranome. It is a collection of paralogous genes. Pseudogenes. These are duplicated genes that have lost their function. Xenologues. Homologous genes within a genome ascribed to the same gene family obtained by horizontal gene transfer. Orphans. Single copy genes without any homologues. Figure here represents the ancestry of orthologs, paralogs, and xenologues. Accumulation of variation occurring in nucleotide sequence helps to infer the evolutionary relationship among the sequences. A gene tree represents evolutionary relationship between groups of sequences. Example in the adjacent figure. A population has an allele, say A1, fixed in the population. Mutation occurs at this locus and it becomes A2. Thus, the particular population is polymorphic for that particular gene loci. Speciation occurs and the population splits. In one population, with time, allele A1 gets lost and A2 gets fixed. Let's name it species 1. In the other population, another speciation event occurs and leads to the formation of two species, say species 2 and 3. If species 2, the allele A1 is lost and A2 is fixed, while In species 3, A2 allele gets lost and A1 is fixed. In this pattern of evolution, in the gene tree, alleles of species 1 and 2 are more closely related, that is, orthologs, than to paralogs gene in species 3. But when we consider the species 2 and 3 are closely related. Hence, a gene tree may not always be similar to the species tree. In the process of evolution, many genes get lost at different lineages. In the process of speciation, variation occurs among population. Natural selection acts on them. With time, certain alleles get accumulated and fixed in the population. This leads to differences among the population over time, resulting in genetic barrier and reproductive isolation. This is when the two population is called a species. Speciation genes can be defined as genes that differ between a pair of species and cause reproductive isolation. It is observed and postulated that the rate of sequence evolution equals the fraction of sites that undergo a change in some designated period of time. This equals the number of replacements per site per million years. Rates of evolution differ from one protein to another. However, it has been found that sex-related genes evolve faster due to sexual selection. In the flowchart here, it illustrates the effect of variations at different levels and the changes it may bring. Example, a substitution can alter the protein expression, its properties and function. This may reflect in the phenotype. The change in phenotype may affect the fitness of an organism in the environment. Remember, the accumulation of favorable genes and fitness of the organism is based on environment and natural selection. This may affect the post-zygotic isolation, both intrinsic and extrinsic. In some cases, the phenotype may pleiotropically affect both fitness and pre-zygotic isolation. Example, in the case of Darwin Finch's beak pattern. We had discussed it earlier. The size of effect may vary from 0 to very large. All these combine to form the total reproductive isolation. A change or variation in a locus may affect the phenotype and reproductive isolation differently. It could be from 0 to large. The figure here represents the lack of correlation between the effect of a locus on phenotype and reproductive isolation. The x-axis here represents the effect of locus change on the phenotype, that is, the morphology, behavior, and physiology, while the y-axis represents the effect of change in locus on the reproductive isolation. The effect of variations can be found by the Dobzhansky-Muller incompatibilities, or also known as the snowball effect. We had studied about DM model earlier. Now, in this case, if you see, in case of Solanum species, a small difference in phenotype but a large difference in the reproductive isolation mechanism. Basically, hybrid incompatibilities. In case of Heliconosus species, A large phenotypic variation as well as a large reproductive isolation is observed. In case of Drosophila cuticular hydrocarbon loci, a small variation in phenotype and also a small variation in reproductive isolation mechanism. While in case of EDA locus in sticklebacks, there is a large change in phenotype or variation in phenotype is seen, but a small reproductive isolation effect. Rules of speciation. Variations are always occurring in nature. Sexual genes and traits are rapidly evolving and generate new variations. This leads to rapid evolution of sex and reproduction related genes, which lead to reproductive isolation. Pre-mating isolation is found to be evolving faster than post-mating isolation. And accumulation of hybrid sterility over hybrid inviability factors are seen. There is always preferential involvement of X chromosomes in hybrid incompatibilities. Speciation genes. These are genes that are responsible for reproductive isolation, a requirement for speciation. Examples, the DSAT2 gene in Drosophila melanogaster. Two variations have been found in DSAT2 gene. The female Drosophila flies from Africa and cosmopolitan have different cuticular hydrocarbons. They vary in a single double bond, but this variation affects the smell of females and affects male choice. Hybrid male sterility HMS gene. This is also identified in Drosophila. It causes sterility in male hybrids formed between a cross between Drosophila similans and Drosophila mauritiana. Example of speciation genes. Speciation genes for hybrid inviability. Example, the receptor kinase 2 gene XMRK2 in platyfish. We had discussed about this while studying the DM model. Second, the hybrid male rescue or HMR gene in Drosophila. This gene is located in the X chromosome. If a cross occurs between Drosophila melanogaster females and its sibling species, it yields dead hybrids. The development is arrested at the larval pupil transition. The sterile hybrid daughters too die when reared at high temperatures. HMR gene also affects fertility of hybrid daughters. The period gene that is seen in Drosophila. It contributes in the difference in courtship song between two species and is associated with behavioral discrimination. The table here represents certain speciation genes that are involved in prezygotic and postzygotic reproductive isolation mechanisms. Thank you.